Okay, thanks. Yes, so well, today I'm going to talk about biogeographic patterns in the Indo-Pacific, and specifically I'm going to look at the influence of life history traits on producing those patterns. Even more specifically, I'm going to be looking at, concentrating on the isolated assemblages within the Indo-Pacific, because I think it's in these assemblages that we see some of the life history influences emerging. So my approach towards this question has been to build a database of life history traits, and so it's something that I've been working with a couple of colleagues, Betty Willis and James Guest, and over the last couple of years we've mined the literature and also done a lot of original research to increase the amount of information we have with a couple of these traits. Most of these traits are traits that you would expect to have an influence on biogeographic patterns. Many of them are likely to have important influences on dispersal in particular. So we've looked at sexuality. Another good thing about corals is that they don't have a lot of traits. They're fairly simple organisms, and so you can categorise a lot of these traits very easily. So they're either hermaphroditic or gonochoic. We're also looking at the mode of larval development, and again, typically only two modes of development. I'll go into this in a bit more detail in the next slide. One other thing we're looking at is the mode of transmission of symbionts. So corals are a symbiotic organism with algae in them, and some species actually put those algae directly into the propagules. That's likely to influence dispersal because they can use the carbon that's produced by the zooxanthellae. And another character trait that I've been looking at is propagule size, and again I'll show you later that size is likely to influence dispersal through its effect on rates of development. So just another plug for the database. I think since the last major review in '99 we've doubled the records, and many of these traits are highly conserved, in particular sexuality. There's a couple of big clades, and so I think we can now make a pretty good guess at the state of these characters for nearly all the scleractinian corals. Okay, so this is a slide demonstrating a gonochoic coral. What that means is each colony only expresses one sex, and so the top is a slide of Lectastria. Again, this is a new record. This guy, people used to think this was a favid. Favids are typically hermaphroditic, and so you can see that the sex is a good character trait for phylogeny as well. So here we can see some eggs in a female colony, and here are spermaries in a male colony. So separate sexes. Here's a hermaphrodite. In this polyp you can see both the eggs and the sperm. Sex may influence patterns of dispersal because hermaphrodites have a slightly higher probability of establishing a new population, given the fact that they can interbreed, whereas a gonochoic species you need at least a male and a female to arrive in a particular location before you can produce some babies. The other trait is reproductive mode. Again, corals typically either broadcast spawn or brood. This is what happens in broadcast spawning corals. The gametes are shed into the water, the embryo develops in the ocean, and so there's an obligate period under which the larvae must stay in the plankton. And so this is some work that I did with Sean Connolly, looking at the competence curves, and you can see that the larvae typically, competence peaks about 7 to 10 days after release. So and they're not usually ready to settle for about 3 or 4 days. So these species have to stay in the plankton for a minimum of 2 to 4 days. Brooders, in contrast, are released. The planular larvae is released fully developed, and it can swim on release. And again, so you can. And he's looking at frequency distribution of settlement. And what you can see here is that 50% of these cohorts of Ceratopora and Stylophora are ready to settle within 6 hours. So the probability of a brooder settling closer to home is much, much higher than it is for a broadcast spawner. This is just a slide to show the difference between vertical and horizontal transmission of symbionts. This is an egg of Montipora digitata, and the little red dots in there are the zooxanthellae. In contrast, this is an Acropora millipora egg and no zooxanthellae. So, and there has been work done to show that, not in this species, but in Possipora damicornis, that the larvae can actually derive nutrition from the zooxanthellae, and so that allows it to be autotrophic and therefore potentially spend long periods in the plankton. So again, you would predict that species with vertical transmission may be able to disperse further than those without. This is just a slide demonstrating the effect of the size of propagules on rates of development. So again, this is data that I've been gathering over a number of years. I think there's about 18 species there, and what I've got here is the diameter of the propagule on release and then the time to motility. And so you can see that one thing, there is a large range of sizes in scleractinia, from about 200 microns right up to 800 microns. And the smaller the egg is, the more rapidly it is swimming. So up here are the acroporas with egg sizes about 500 microns. It takes them 30 or so hours before they even begin to move. Whereas a Possipora larvae with an egg of about, a Possipora egg of about 100 microns is swimming within about 10 hours. So again, you would predict that egg size might have a strong effect on patterns of dispersal. And so for the rates of development, I categorise the eggs into 6 size categories, and all brooded larvae are recorded I classified as category 1, meaning that they are likely to spend less time in the plankton because they're ready to settle on release. And so another couple of sources of data to do the study. So we are working with one of Terry's research assistants. We're using Terry's data on species distributions of corals. We chose 51 islands, and we looked at islands with a range of different isolation indexes. There's a UNEP page which lists all the islands in the Indo-Pacific, all the islands in the planet, and it gives you an index of isolation for that island. We modified the index slightly. It's based for terrestrial systems, and so they record the distance to the nearest landmass. We changed the index to measure the distance to the centre of diversity, and so then we combined these 3 sources of data to look at the relative abundance of species in these 51 assemblages. And first of all, this just shows the pattern that Sally put up yesterday, just to show how we've captured isolation is a very strong driver of species richness. So basically this is just saying the further you get across the Indo-Pacific, the less species you get. So basically just to see that this isolation index was capturing the broad biogeographic patterns. The other thing we looked at was the proportion of endemics in the assemblage. So here is that marine isolation index, and this is the proportion of endemics in the species. So what we're seeing is that there's a very constant proportion of endemics for islands over a broad range of isolation, and then you seem to hit this point beyond which the proportion of endemics here is almost doubling. So there seems to be some threshold distance of isolation beyond which we might expect to see some influence of life history. This is the proportion of the coral assemblage which broadcast spawns. Again, not many patterns here. The R squared is very low. Typically a very constant, about 97% of the species broadcast spawners wherever you go in the Indo-Pacific. A few outliers, but so no patterns in terms of the mode of larval development. The number of hermaphrodites, the proportion of hermaphrodites in the assemblage, again, very constant until you hit this isolation index of around 150, when things start to change. I'm working with Josh Madin at the moment to put some models to these and try and get some statistical values to see whether these things are, whether what we're seeing here is not just by chance. Again, the proportion of the coral species with symbionts in the propagules. So again, for most of the islands that we looked at, a constant proportion of about 20%, again, until you hit this threshold, above which the number of the proportion of species in the assemblage with symbionts in the propagules nearly doubles. This is just another way of looking at that. This is another data set that's just become available from Charlie Veron, has made all his data available online, and he's classified something like 135 regions, and so what each one of these dots represents is one of Charlie's regions, and the proportion of zooxanthellate larvae in that in those assemblages is proportional to the diameter of these circles. And again, so you can see that here in the centre of the Indo-Pacific, it's a fairly constant, and it's all these peripheral locations have an overabundance of species with symbionts in their propagules. This is looking at the effect of the relative abundance of species with the different egg sizes or rates of development. And again, a very similar pattern here. We've got the actual islands listed, but they are ranked in order of increasing isolation. And so again, you can see that the relative abundance of species with different egg sizes stays very constant until we get to these really isolated islands, when it starts going all over the place. So, just to summarise all that, so there seems to be a breakdown in these assembly rules beyond a given threshold of isolation. So for most of the islands that we look at in the Indo-Pacific, the relative abundance of species with different traits is very similar, and that you see that in the community structure as well. The relative abundance of species in different families is very similar as well. But that breaks down at a given threshold of isolation, and beyond that threshold, what you see is an overabundance of species with rapidly developing propagules and an overabundance of species with zooxanthellate in their propagules. So what I think this says about the influence of life history traits on biogeography, I think it suggests that the good colonisers need two traits. So there's no one particular trait that's going to determine distributions. So they need a combination of traits. And that combination of traits is autotrophic larvae. So the species with autotrophic larvae can stay in the plankton for long periods of time and make these transoceanic journeys and arrive in a new location. But once they get there, to establish a population, they need to have a propagule that's likely to be retained. And so it's those species that have small eggs or are brooders that can both arrive at these locations and then establish a population. And I'll end there.